So far, we have covered the chemical structure of DNA, the structure of the nucleotides, how the nucleotides come together, hydrogen bonding, covalent bonding, and ultimately that double helix twist that we now know is our overall structure of DNA. But DNA takes on even more intricate structures. And the reason for this is that DNA has to pack into little tiny cells. For example, in E. coli, DNA, if you were to stretch it out, is about 1,000 times longer than the cell itself. And when we look at eukaryotic chromosomes, for example, in humans, our smallest chromosome is 14,000 times longer than the cell. So you have to get what is essentially a really big molecule into a little tiny cell. And the only way to do that is to pack it perfectly, to pack it down tightly so that you can compact it into the cell. And DNA is packed by supercoiling. So supercoiling is a twist that then continues to twist on itself. So I always think of coiled telephone cords, which just says that I'm getting old. But if any of you happen to remember a coiled telephone cord, you probably remember that over time the coiled telephone cord would twist up on itself. So you would have this coiled cord, and then eventually it would sort of twist back, and it always got a little shorter. And sometimes it would even twist back even more and make a little knot. So our cells are doing essentially the same thing with our DNA. And you can do the same thing if you grab a rubber band, stretch it out just a little bit so it's flat instead of a circle, and then twist one end while you hold the other. And you'll start to see the double helix coil. And if you keep twisting that rubber band, it will begin to supercoil. It will begin to coil back on itself. And you'll notice that if you do it slowly and evenly, it actually makes really nice, neat coils. So our cells are doing the exact same thing. On the right-hand side here, we have a circular piece of DNA. And if you were to turn it, a right-hand turn, because our helix is right-handed, so we have a right-handed helix. And if you continue to twist it in a right-hand direction, your twist will begin to twist. So a positive supercoil is a supercoil that's going in the same way as the helix, and it just twists back on itself. Now, if you have a coiled molecule and you turn it the opposite direction, you are going to uncoil it. So a negative coil is going to open up the molecule. First, it will unwind the supercoil, and then it will actually open up the DNA molecule. So the negative is moving oppositely against the helix. Positive is moving in the same direction as the helix, essentially to make it tighter. If you go into Brightspace, there is a video linked on supercoiling that just kind of shows what happens in 3 dimensions. It may not be this exact video that's linked here. That's okay. You can watch it, whatever is linked in Brightspace. Occasionally I have to change them out. Or you can go back to the lecture notes and click on this one. In the PDF, you should be able to click on this one. So our positive supercoil is going to tighten up and compact our molecule. So it tightens and it compacts. And our negative supercoil is going to loosen and potentially undo the helix. Now, the enzyme that is responsible for this is called topoisomerase. And we're going to see topoisomerase when we cover DNA replication in chapter 9. Topoisomerase is one of our DNA replication enzymes. And its job actually is untwisting the supercoiling ahead of replication. But it doesn't just work in DNA replication. It also helps fold the DNA nicely and neatly into the cell, either by a positive or a negative supercoil. So our bacterial chromosome is a single circular piece of DNA. This DNA appears to clump in the nucleoid region. Remember, the nucleoid is a region, not an organelle, so there's no membrane surrounding it. Bacterial DNA is twisted into supercoils and is generally sort of maintained by special proteins that just kind of keep it nice and neat. When we get to eukaryotes, we're going to learn about histone proteins. So bacteria don't get complexed around histones, but there are proteins that sort of help with the organization. We'll look at histones in just a few minutes. But we can see this picture here. There's these little proteins that are just sort of keeping the molecule organized, not necessarily in a specific pattern, but just kind of generally packed into the cell. Our eukaryotic chromosome is a little bit different. So the eukaryotic chromosomes, because they're so big and because there's so much DNA, we have to package them even more precisely. And so I want you to think back to when we learned about our karyotyping. And remember that karyotyping is imaging chromosomes using specific stains. Now, there are different stains that will stain DNA molecules differently in different regions, but many of them will stain regions based on how tightly they are packed. So if we look at this chromosome, we have regions that are light and regions that are darker. Those light regions are called euchromatin, and these are less condensed regions. Now, we'll tend to see these regions on the arms of the chromosomes as opposed to the centromeres or the telomeres. Except for this one, we've got clear or light ends on this one. But generally, you're going to find the telomeres are extra densely packed and the centromeres are more densely packed. So we look at the center of this one here, we can see that densely packed centromere region. So our less condensed euchromatin are going to be regions that have more genes. And the reason that it is less condensed is that it has the DNA polymerase, RNA polymerase, I'm sorry, not DNA polymerase, RNA polymerase needs to get to those genes. So if we have lots of genes, they're probably going to be regions where we're doing a lot more transcription. And so the cell is going to maintain a less packed state, just so that RNA polymerase can get in there. Our highly condensed regions are called heterochromatin. These are regions like the telomeres and centromeres where there are relatively few genes, so transcription doesn't take place there very often. So the chromosome can sort of maintain a tighter packing there. In order to condense, our chromosomes are wrapped around these histone proteins. So histone proteins basically act like little spools around which DNA is wrapped, and they are going to be the basis for our folding and our packing of our molecules. There are 4 different histone proteins that are present in our cells. There are 2 copies of each of these histone proteins to make 8 proteins all together in the histone complex. Histones are positively charged. This is because they are interacting with DNA. And remember, because of our phosphate groups, DNA is highly negative. So if something's going to interact nicely with DNA, it's got to be positively charged. So our histone proteins have that positive charge so that the negative phosphate groups are attracted to it, and that's going to make coiling around them even easier. So our chromatin structure is DNA, the DNA helix, plus the associated proteins, in this case, our histone proteins. So our chromosome overall is going to be our DNA molecule plus these DNA and protein regions. Our lowest level of DNA packing is called the nucleosome. A nucleosome is DNA wrapped around histone proteins, and it wraps around roughly twice. So we can see down here on the bottom, we've got our histones, and then we've got our DNA molecule wrapped twice around it. So let's actually go this way. Whoop. I don't understand that coil. Anyway, that's just the drawing. Don't worry about that too much. In between our nucleosomes, we have this region of DNA called the linker DNA. So we have DNA wrapped, a little space, DNA wrapped, a little space. So the H1 protein, the histone 1 protein, is not shown here. It's on the next slide. It is going to attach to sort of the outside edge of our nucleosome. And then we have 8 other proteins, histone 2A and B, histone 3, and histone 4. And there are 2 copies of each of those to make up our histone. So I want to draw your attention to the center of this image really quickly. This yellow protein here is the H1 protein. So H1 is our yellow orangey protein. So H1 histone 1. And then we've got these purple proteins in here. These are the H2A, H2B, H3, and H4 proteins. We've got our linker DNA in between, and we've got our double helix wrapped twice around those histones. So that is going to be the first level of packing after our helix. So if we start up at the top, we've got our DNA helix, remember, made up of our nucleotides. So nucleotides are going to be primary structure. Then DNA is going to wrap around on itself. The double helix is going to be that secondary structure, or the first level of sort of 3D structure. And then after that, we're going to begin our packing. So that first level of structure is going to be the nucleosomes. And then after that, our chromosome starts to pack more and more tightly. So think about twisting that rubber band and continuing to twist it. And if you do it slowly enough, it's going to twist back on itself. So what we have first are these nucleosomes kind of wrapped tightly, wrapped really closely together, sort of twisted together. And we call this a 30 nanometer fiber. So our twisted nucleosomes are going to be the 30 nanometer fiber. It is called the 30 nanometer fiber because when you measure it using X-ray crystallography, it is 30 nanometers tall, which is a very tiny distance. And then our chromosome continues to fold and twist, and it twists into, or folds into, this sort of twisty, turny, almost accordion-like shape. And that averages 300 nanometers across. So our width here is 300 nanometers. So we call this the 300 nanometer loop. Then we can continue twisting and turning, and we get a little bit thicker. We're getting more condensed, but we're getting wider. So we're getting shorter, but we're getting a little bit wider. We continue to do this until we get to the 1400 nanometer chromosome. This is our metaphase chromosome. So we are familiar with this guy. This is 2 sister chromatids that have been produced by DNA replication. And this average distance is 1400 nanometers. So our most tightly compact and condensed state in which we will find our chromosomes is in metaphase. And we want that because we want our chromosomes packed nice and tightly so nothing breaks. But as we go through replication and transcription, all of this unpacking is going to have to relax to allow our transcription and replication enzymes to actually access that DNA molecule. So when gene expression or DNA replication is not happening, our chromosome is going to stay more condensed. It's not going to stay as condensed as this 1400 nanometer metaphase chromosome because we only need it that tight occasionally. But our euchromatin regions are going to be more relaxed than our heterochromatin because the DNA molecule has to open up more frequently for transcription. Again, there's a lecture video linked here. If you go into Brightspace in the additional resources folder, you will see a variety of different videos there that you can watch. All right, so our chromosome structure is not only important for the physical packing, but also for a little bit of gene regulation. So changes in our chromatin structure and our chromatin packing can actually result in changes in gene expression. And physical changes, such as changes in chromatin structure that affect the expression of genes, are called epigenetic changes. So we'll talk about these a little bit later, but one example of an epigenetic change is methylation. So a methyl group is a CH3. And sometimes the cell will methylate the DNA, add a methyl group to the structure. So this is not changing a nucleotide. This is not changing any bases. This is adding a methyl group to the base. And what this is going to do is change the ability of RNA polymerase for transcription to get into that gene. And so you might see a gene and expect a particular phenotype, but it may not be expressed because it has been methylated and shut off. So sometimes this is permanent, sometimes this is reversible, depends on the gene, and it depends on what's causing the methylation. So sometimes environmental factors will cause methylation to happen. Sometimes demethylation will happen. The other thing that's interesting about epigenetic changes is that they can be heritable. So if a gene has been methylated in one of the parents, say the mom, it may remain methylated in the child, causing a change in gene expression. Our example here is agouti mice. Agouti is sort of this speckled black and brown color. You can see we've got a dark mouse and a light mouse. And then these ones in the middle have sort of this spotty black and brown coloring. That is the agouti coloring. So DNA methylation at the agouti gene actually produces different types of gene expression. So we might get more or less dark colored fur compared to an organism that has not been methylated. All right, so we talked about how we get more heterochromatin at the telomeres, the ends, and the centromeres. So the centromere, we've talked about that quite a bit in mitosis and meiosis. This is a really highly condensed region of heterochromatin. This is where the spindle fibers attach. So remember that we talked about the kinetochore, and the spindle fibers actually attach at the kinetochore, which is essentially a band of proteins around the centromere. So the spindle fibers are not actually binding to the DNA itself, it's binding to the kinetochore, which is kind of like a belt around the centromere. But the DNA molecule itself is highly condensed there. And then we have the telomeres. So the telomeres have a really unique purpose. The purpose of the telomeres is basically to protect the ends of the chromosome. And we'll come back to telomeres in chapter 9. and talk about what happens at the end when we replicate the ends of the chromosome. But basically, the gist of this is that when we replicate the ends of a chromosome, we lose a little bit every time. And this has to do with leading strand and lagging strand replication, which we, again, will talk about in chapter 9. But every time we replicate a cell, replicate our DNA in a cell, we lose a little bit of the end. Telomeres are repetitive regions that act as buffers so that when this process of replication happens and we lose a little bit of the end, we don't lose anything important. But telomeres also act as a cap. So what happens is that we have a region at the end that is relatively GC rich. Remember, G's and C's make 3 hydrogen bonds, so they make a sort of stronger bonding region than an AT rich region. And so what happens is this overhang on the end will loop back and pair with a complementary sequence a little ways down the chromosome. And what it does is instead of leaving the end free, it makes a loop or a cap on the end, which is basically protecting this DNA strand from degradation, so that the ends of the DNA molecule are protected. We also have variations in different regions of our genomes. And then genomes vary in general. So genomes vary by size of the organism, although size is not necessarily related to complexity. There's no sort of linear increase in DNA. Plants tend to have a bit more DNA than animals do, kind of in general. Not that that is necessarily reflected here, but plants do tend to have much larger genomes, a lot of extra DNA. Our viruses and bacteria, because they are small, are always going to have smaller genomes. And this is basically a space thing. So our E. coli isn't going to have a lot of extra DNA that's not doing anything. If it's not being used, it's probably going to be removed from the genome. But our eukaryotes are much bigger cells and they can handle more genetic material. So you can see some examples here. But again, the difference in complexity doesn't really have anything to do with difference in genome. So for example, we've got this salamander down here who's got 765 billion base pairs compared to humans who have 3.2 billion base pairs. And then you can just see some of the other variations. So there's not a clear pattern in genome size. So to wrap things up, wanted to talk just briefly about the actual sequences we find in our genome. So there are a few different types of sequences that we're going to see. There is unique sequence DNA. These are going to be coding sequences. And when we say coding, we mean that we're going to produce a protein from it. And that protein is going to have a function in the cell. These might be similar because we can have groups of genes that are evolutionarily related, genetically similar, and that's perfectly fine. But they're going to do something different. They're going to make different proteins. We might also have non-coding regions. So these are regions that are usually going to make RNA molecules. So we're going to get an RNA molecule from our DNA, but they don't produce a protein. Instead, most of these RNA molecules have some sort of function. A lot of them work in regulating gene expression, and we'll talk about some of these a little bit later, but they do jobs in the cell. And our telomeres fall into this non-coding region. There are other regions that have structural significance, sequences that aren't coding for anything, but are helping to maintain the structure. Maybe they're there for producing heterochromatin or something like that. And then lastly, we have repetitive DNA. Repetitive DNA comes in different forms depending on how long the sequence of repeats happens to be. Not the actual repeats themselves, but how many repeats we get. So we have different types of repeats. So we have tandem repeats, which are 2 base pair repeats. So this might be like GC, 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 etc. For some number, some length, maybe 300 base pairs, we would get GCs. We also have what are called interspersed repeat sequences. These are short sequences that are interspersed by other sequences of DNA and long sequences that are interspersed by other pieces of DNA. And then we have highly repetitive DNA. These are usually short sequences, so maybe less than 10 base pairs, but we have many, many, many copies. So some of these are helpful, like we might use SNPs, which are single nucleotide repeats. Regions that are full of single nucleotide repeats might be used for RFLP, which was that restriction fragment length polymorphisms, or DNA fingerprinting. So we can use some of these regions for different reasons. We'll cover some of these in some of our biotech. Others, we won't really say anything else from here.